Everybody, and welcome to Mr. Saucedo's YouTube videos. Today we're going to be talking about the structure of DNA. So, we have been slowly but surely covering all the characteristics of living things that we talked about in Unit 1. And this one is all about living things having universal genetic code, aka DNA. So, we're going to kind of talk through an experiment that was done in the 1920s by a gentleman called Frederick Griffith. And so hopefully we'll be able to kind of come to some sort of conclusion like he did. Okay, so we're just going to kind of use the information that was given, that is given to us by his experiment. So up until that point, we weren't really sure what, I know it sounds stupid. Like everybody knows DNA is what's in control of what we do and what our cells do, right? But no one was really sure in the 1920s. It could be proteins. Are proteins the information carrying molecule? RNA, DNA. There are a lot of things that could carry information around our cells, and we weren't really sure which one it was. So here's kind of what his experiment was. So he took some bacteria, and there are two different versions of the same type of bacteria. This one, it says rough strain because it under the microscope it looked rough. Smooth strain looked smooth. Go figure, right? So you can think of this as like R and this is S. The R strain doesn't kill you. The S strain gives you pneumonia and you die. Okay? They're both a form of streptococcus, but this one doesn't really give you any symptoms or anything, and you are totally okay. This one, if you're given it, you get pneumonia and you die. So he was injecting mice with it, and so like you can see, like this makes sense. This one's not supposed to kill you. This one is. So what he decided to do though was, okay, I'm going to take the bacteria and I'm going to kill it. So I'm going to kill it with heat. So heated it up until they all were dead. I'll inject that into a mouse. What do you think is going to happen? Answer: the mouse lived. Okay. Then he took the non-virulent kind, the one that isn't supposed to kill you, and he took the dead smooth strain bacteria. He injected a mouse with that. What do you think happened? The mouse died. The question is why? Right? And this is the 1920s that we're talking about here. But why? So, Frederick Griffith in 1928 discovered bacteria transfer some kind of genetic information to each other. Even the dead ones, the living ones were able to take information from the dead ones. And that was able to change them somehow, or they were able to utilize that in order to basically make themselves deadly. Okay, what was it that they were transferring, right? So, hypothesis here. It's either going to be protein, or it's going to be DNA, or it's going to be RNA. Those were the only three molecules people could think of that could possibly be carrying this genetic information. Okay? And so what do you do? Well, we have enzymes, remember, that can destroy things and can break things apart. So there is a proteinase that gets rid of protein. So there's no protein in a sample. There's RNase, which gets rid of, you guessed it, RNA. So there's no RNA in a substance. And then we have DNA. So DNase. You can get rid of DNA, destroy it, so that there's only, RNA and protein left. And so that's kind of what we used. And so we said, great, so let's inject them with we're repeating the experiment again, okay? But we add proteinase first, and then we inject it into the rat. What happens to the rat? It died. Same exact experiment. Now there's no RNA. What happens? Dies. We add DNase. Destroy the DNA. Then we inject it into the mouse. It lives. So, 1944, this is like 20 years later, Oswald Avery, this guy, discovered that the genetic information that was being transferred between the bacteria was DNA. That must mean that DNA is the information carrying molecule inside of bacteria cells. So conclusion, DNA stores and transmits genetic information, just like passing the baton. So we went from Frederick Griffin and we went to Oswald Avery, passing the baton to the next generation. Okay? So we want to know more, though, about DNA. So this is called the Hershey Chase experiment. 1952, so not that long after Oswald Avery's discovery, Alfred Hershey and Martha Chase decided to prove that DNA was the only carrier of genetic information. Okay? So all that we knew was that, great, DNA carries genetic information in bacteria. And that based on the experiment, that seemed to be the only thing that was doing it. But that doesn't mean that it is the only carrier. There could be a whole bunch of other ones. Maybe RNA did play a role, but just not in bacteria. Or maybe, I mean, there are plenty of different ideas out there. And so what they did was they used viruses that infect bacteria. They're called bacteriophages. And they decided to use radioactive dye. Again, we're in the 1950s here. So radioactive dye was a very new thing. And they waited to see whether viruses injected DNA or RNA or proteins. And the nice thing is DNA, RNA, and protein, they look different if you dye them with, the proper isotope that is radioactive. So what they found, right, is that this big old glowing bit that's in here is being injected directly from the virus into the bacteria. And they found that it is only DNA. RNA is never transferred. Protein isn't transferred. It is literally pure DNA that is going from the virus into the bacteria, infecting it. And so that's called the Hershey Chase experiment. So structurally, what are we talking about when we say DNA? We're talking about deoxyribonucleic acid. The D stands for deoxyribo, the N, nucleic, A, acid. So DNA is made up of a lot of different things, but nucleotides are the primary subunit that make up DNA. And so nucleotides contain the following. They contain a phosphate group. That's supposed to be this little circle here, a 5-carbon sugar. In our case, for DNA, it is deoxyribose sugar, and a nitrogenous base. So that's what we're really looking at here. Okay? Now, there are 4 types of nitrogenous bases. You probably are aware of them, and you've probably seen them before, but you can see this is our phosphate, this is our deoxyribose, and then this is supposed to represent one of the nitrogenous bases. We have adenine, A, thymine, T, cytosine, C, guanine, G. And so that's what these are supposed to represent. And maybe you forgot this, maybe you didn't from first semester, but we talked a little bit about this already. These little dashed lines are supposed to represent hydrogen bonds that are holding the structure of DNA together.